Hello friends, welcome back to my YouTube channel Journey with VIJAYKUMAR SHRIVASTAV. Today my presentation title is Vernalization. Before presentation, little introduction about myself. I am VIJAYKUMAR SHRIVASTAV. I have done M.Sc Agriculture with specialization in Agronomy and presently working in seed industry and associated with one of the M.Sc. So let's move to the presentation. What is vernalization? The cold treatment given to plant buds, seeds or seedlings for promoting early flowering is known as vernalization. In short, the chilling treatment for induction of early flowering is called vernalization. Vernalization is the requirement of plants to go through a period of cold before they can flower. Besides an appropriate photoperiod, certain plants require a low temperature treatment during their early stages of the life for subsequent flowering in the later stages. This low temperature treatment requirement was termed vernalization by Lensko in 1998. Due to vernalization, the vegetative period of the plant is cut short, resulting in an early flowering. In nature, vernalization takes place in the seed stage in annuals like winter rye. The biennials and many perennials respond to cold treatment at a very late stage like crops, henbane and apple. Now to study about perception of cold stimulus and presence of floral hormone. The cold stimulus is perceived by the apical meristems. The perception of cold stimulus results in the formation of a floral hormone which is transmitted to other parts of the plant. In certain cases, the cold stimulus may even be transmitted to another plants across a graft union. For instance, if a vernalized henbane plant is grafted to an unvernalized henbane plant, the latter also flowers. This is due to the induction of the plants to produce a hormone named as vernalin by Melchers, described in 1939. Techniques of vernalization. In this technique, the seeds are first soaked in water and allowed to germinate in processing chamber at 10 to 12 degrees centigrade temperature. Then the seeds are transferred to low temperature 3 to 5 degrees centigrade and kept for correct period of time, which varies on different varieties or few days to 30 days. Then seeds are dried and sown in the field in proper oxygen and water supply conditions. Now we will go through the conditions which are necessary for vernalization. First one is age of the plant. The age of the plant is an important factor in determining the responsiveness of the plant to the cold stimulus, and it differs in different species. In cereals like winter wheat, the vernalization is effective only if the germinating seeds have received cold temperature treatment for sufficient time. While in case of biennial variety of henbane, the plant will respond to cold treatment only if they are at rosette stage and completed at least 10 days of growth. Here to study the relationship between vernalization and age of plants. In cereals, vernalization best occurs in germinating seeds and even at embryonic stage in mother plant. As per opinion of Sarkar, Hyoscyamus niger gets maximum sensitivity to chilling effect after 30 days of growth, and in Enothera, maximum sensitivity to low temperature occurs only when the plants bear 6 to 8 leaves. Second condition, appropriate low temperature and duration of the exposure. Most suitable temperature for vernalizing the plants ranges between 1 to 6 degrees centigrade. The effectiveness of low temperature treatment decreases from 0 to 4 degrees centigrade. Low temperature at about -6 degrees centigrade is completely ineffective. Similarly, at high temperature from 7 degrees centigrade onwards, the response of the plants is decreased. Temperature of about 12 to 14 degrees centigrade is almost effective in vernalizing the plant. Besides an appropriate low temperature, a suitable duration of the cold treatment is essential for vernalization. Depending upon the degree of temperature and in different species, this period may vary, but usually the duration of the chilling treatment is about 1.5 months or more. Third condition necessary for vernalization is oxygen. The vernalization is an aerobic process and requires metabolic energy. In the absence of oxygen, cold treatment becomes completely ineffective. And fourth condition, water. Sufficient amount of water is also essential for vernalization. Vernalization of the dry seed is not possible. Here we will study about mechanism of vernalization. There are two theories. First one is hypothesis of hormonal involvement and second is hypothesis of phagic development. In mechanism of vernalization, first we will go through phagic development theory. This theory was proposed by Lysenko in 1934 and it describes as first the growth which is increase in size and development which refers progressive change in the characteristics of new organs are two distinct phenomena. Second, according to this theory, the process of the development of an annual seed plant consists of a series of phases which must occur in some predetermined sequence. Third, commencement of any of these phases will take place only when the preceding phase has been completed. And four, the phases require different external conditions for the completion such as light and temperature. And fifth, vernalization accelerates the thermophase like that phase of development which is dependent upon temperature. Thus, in winter wheat, low temperature is required for the completion of first thermophase. After this, the next phase that is dependent upon light known as photophase starts. Vernalization of winter wheat accelerates the first thermophase so that there is an early swing from vegetative to reproductive phase or flowering. In hypothesis of phagic development, the development of annual seed plant consists series of phases which occurs in predetermined sequence. Next phase only initiates on the termination of the previous phase. There are two phases. First one is thermostage and second is photostage. First one is thermostage. It is vegetative phase which requires low temperature 0 to 14 degrees centigrade and suitable moisture and aeration. This stage is of variable length and depends on the nature of plants and environment. Second phase is photostage. This phase requires high temperature and here vernalization helps in synthesis of florigen. Winter wheat completes their life cycle more rapidly when given a short day and low temperature during thermostage and a long day and higher temperature in photostage. The second mechanism of vernalization is hormonal theories. It has already been described that vernalization probably involves the formation of a floral hormone which is called vernalin. Based on this fact, many hypothetical schemes have been proposed by different workers from time to time. The first hormonal theory proposed by Long and Melchers in 1947 and which has been shown is described in the diagram. According to this scheme, the precursor A is converted into a thermolabile compound B during cold treatment. Under normal conditions, B changes into C, which ultimately causes flowering. But at higher temperature, B is converted into D and flowering does not take place, which is known as devernalization. In this diagram, we have shown the relationship between vernalization and flowering. If the high temperature is given, it will convert to devernalization. Now we will study hypothesis of hormonal involvement is proposed by Chalakyan. Vernalin is produced in some biennials, winter cereals, and perennials like rice, anthemum, and apple at low temperatures. In long day plants, vernalin is converted into gibberellin. Anthocyanin is present in long day plants. Anthocyanin along with vernalin cause flowering in long day plants. But in short day condition, the vernalin is not converted to gibberellin because they lack anthocyanin, hence flowering does not occur. Devernalization. The positive effect of the low temperature treatment on the vernalization of the plant can be counteracted by subsequent high temperature. This is called devernalization. The devernalized plant can again be vernalized by subsequent low temperature treatment. Vernalization and gibberellins. The gibberellins are known to replace the low temperature requirement in certain biennial plants such as henbane, where the plant normally remains vegetative and it retains its rosette habit during the first growing season and after passing through the winter period flowers in next season. The gibberellins cause such plant to flower even during the first year. Now we will study the significance of vernalization. First, vernalization shortens the vegetative period of the plant. Second, it increases cold resistance to the plant. Third, vernalization increases the resistance of plant to fungal diseases. Fourth, it is a physiological process that substitutes or compensates the effect of thermophage. Fifth, in biennials, vernalization induces early flowering and early fruit setting. And sixth, a non-vernalized shoot apex can be introduced to flower by grafting the plant with a vernalized plant. So this was all about vernalization. Now my presentation ends here. Thank you very much. Here I have given my YouTube channel details, Journey with VIJAYKUMAR SHRIVASTAV. Having request, please visit the channel, subscribe it and provide your kind and valuable feedback. Thank you.